Hello everyone, my name is Matt and I'm going to walk you through a brief presentation that highlights some of the key points from my poster presentation. So let's get started. I meet our research organism, the northern red-bellied cooter. As its name would suggest, this turtle is known for its bright red pattern on its plastron or its belly. If you were to take a walk around Lake Fred in the spring or the summer, you're sure to see these guys poking their heads out of the water or see them basking up on the shore with their bright red bellies. Now, why study turtles? Number one, I like turtles. Number two, as I mentioned in my poster, there is a clear bias in terms of immune research that favors the study of endotherms over ectotherms, which leaves us with a gap in the literature as far as the relationship between temperature and immune performance. The other reason to study turtles is that turtles and other reptiles and amphibians are in a state of global decline. Uh, Things like pollution, invasive species, habitat loss and degradation, unsustainable use are all contributing to these dwindling numbers, but the two factors we're going to focus on are climate change and disease. Climate change, while you may assume global warming would be good for cold-blooded animals, less time basking, they're just naturally warmer. Climate change is actually very harmful. Studies have shown that over millions of years, ectotherms have proven to be much slower to adapt to environmental and climate changes than their warm-blooded counterparts, leaving them particularly vulnerable to climate change. And disease, a great example of how disease can affect a population of reptiles in real time would be the picture shown on the right. This is a new disease. These are mysterious lesions of unknown origins that have popped up on red-bellied cooters found in Salem County, New Jersey. Uh, my research mentor, Dr. Lynn, and I were fortunate enough to be a part of the New Jersey Fish and Wildlife's efforts to sample these turtles and figure out what's causing these lesions. So it was very interesting to see how disease can affect a turtle population in real time. The goal of this experiment, we want to figure out how does temperature affect in vitro innate immune performance in the red-bellied cooter. Our hypothesis is that turtles will perform best at temperatures that are closer to their ideal temperatures. And our prediction is that if our hypothesis is true, turtles that are incubated between 30 and 40 degrees Celsius will kill more bacteria than those that fall out of this range. As we move into our methodology, sampling turtles, this was the most fun part of this experiment. It was basically just throwing waders on, getting out in the muck of Lake Fred and coming up with a handful of turtles via hand capture. We then took a blood sample and we isolated the plasma back at the lab for use in our bactericidal assay. We used the plasma from 6 turtles in this assay, with 12 microliters of plasma from each turtle going into each temperature. Each of the samples was spiked with 20 microliters of PBS containing a known quantity of E. coli to initiate the assay. The assay was then incubated for 45 minutes to allow bacteria to grow and for the turtle plasma to kill the bacteria before the reaction was halted by putting it on ice. After the assay was complete, we played the results on 2 different nutrient agar plates, and after 2 days, I counted the colonies on all 148 plates, which was definitely the least fun part of this experiment. Statistical analysis, we calculated the BKA score from the colony numbers. BKA score is real simple, as you can see there from the equation. BKA score closer to 1 indicates better killing ability, and a BKA score closer to 0 indicates poor killing ability. We then generated something called a thermal performance curve for each individual turtle, and we also calculated an average thermal performance curve from the least square means of all 6 turtles. We then performed a repeated measures de novo test and a 2-piece post hoc test to check for statistically significant differences between turtles at the different temperatures. And these are our results. You can see each individual curve on the graph on the left. You can see there's some individual variation from turtle to turtle, but they're all roughly the same shape. You can also see the average thermal performance curve on the right. From this data, we were able to figure out that the peak performance temperature is 32.8 degrees Celsius, and the 80% performance breath ranges from 25.2 degrees Celsius to 40.4 degrees Celsius. If you don't know what 80% performance breath is, it's just a range of temperatures at which 80% of the maximum killing ability is achieved. What do our results mean? Well, they mean our hypothesis is partially supported. Like we expected, turtles at lower temperatures did not perform as well as turtles in this optimal range here from 25 to 40 degrees Celsius, but we did not see the expected drop-off here at 45 degrees Celsius, as there's no difference between any of these over here. I came up with a possible explanation of the fever response because a coordinated immune response includes the fever response, which is an increase in body temperature. Perhaps the complement proteins in the plasma have to be able to work well at higher temperatures, but this is just speculation. We can also look at the difference between turtles and snakes in terms of their thermal immune performance. Our lab has done a lot of research with snakes, and something that we found was that turtles required 6 times as much plasma as snakes in order to trigger a strong immune response. We also found that turtles have a wider 80% performance breadth than snakes. What this could mean is that snakes have a stronger immune system, but turtles are the more generalist species, able to perform at a wider range of temperatures, but more research is needed on this. Speaking of that, for future research, I think step in the right direction will be increasing the sample size beyond just 6. It could also be helpful to look at seasonal comparisons. We got the turtles right as they were going out of a hibernative state called brumation. It would be interesting maybe what their thermal performance curves look like going into hibernation. As I just said, we could also do a cross-species comparison, look at a bunch of different reptiles and ectotherms, see how turtles stack up to snakes or lizards. And I think the most interesting thing would possibly be doing a healthy versus infected turtles by taking our healthy turtles at Lake Fred and comparing their thermal performance curves to the samples obtained from the diseased turtles that I mentioned earlier found in Salem County. I feel like that would be a very interesting comparison. All right, that just about wraps it up. So stay healthy, stay safe. Thank you for your time.